break is almost over, so you're skipping the tea break maybe. Okay. What I will share with you is the data which we are collecting at CBR in the context of aging brain research. Center for Brain Research is maybe a few hundred meters away from this complex. So our center addresses the problem of dementia, and WHO has actually in their research, the Blueprint for Dementia Research in 2022 released this document, basically identified as one of the great health challenges of our generation. And we need to come together to address this. What is dementia? Well, the most common types of dementia, what we know is Alzheimer's disease. So dementia is a collection of a group of diseases. Alzheimer's disease is basically almost 60 to 80% of the disease is classified under dementia. Then you have other diseases, and of course you have Parkinson's disease and So what exactly is brain aging? So all of us will age as we, so the body ages, the organs age. So the brain aging also has a certain, let's say, the curve along the x-axis, which is time, and there is a cognitive decline. So healthy aging is the top line, where there will be cognitive decline as time progresses. But when you have a disease like Alzheimer's, etc., the curve, the steepness of the curve increases dramatically. So you can see that AD stands for Alzheimer's disease. There is a term called MCI, which is called mild cognitive impairment. So the pre-dementia phase is called mild cognitive impairment. And it's sort of like the knee of the curve. But what we need to figure out is, if somebody is identified early enough as maybe leading towards that MCI, there is a possibility for us to alter the curve, a steep decline, into a slanting line like parallel to the first one. So the goal therefore is to understand whether we can detect MCI. But there is no cure for Alzheimer's disease as of now. If we can detect MCI, then we can halt the red path, the red part of the curve. Okay, so mild, moderate, severe, and so on. So the manifestation of Alzheimer's disease or other neurodegenerative diseases in the elderly basically happens from a clinical point of view, where a neurologist will tell you or tell the patient that you have these symptoms, and therefore maybe the disease is in a certain stage of progression. So the goal is therefore to understand MCI and see whether we can detect MCI. So how do I do that? It is a process where we need to collect data. So I'll quickly present the case of the challenge of dementia in India. There have been many longitudinal studies done across the country at different points in time. So there's a 1998, 1996 study, then 2013, and so on and so forth. Different parts of the country have different rates of people under dementia. So there is a need for us to understand, because from a social context, population context, how does this disease manifest itself? And this disease actually takes about 15 to 20 years. It starts off about 15 to 20 years ahead in time, but the clinical symptoms will manifest only after a long period of time. So if we can find out what is happening early enough, then maybe we can tackle that. So there is a need for longitudinal studies. So that's what we will look at. So the mission of our center is basically to develop a composite biomarker for detection of the early onset of dementia and design interventions to prevent or postpone dementia. This is our clear focus of what we do. So the key mission points for us is basically we need to collect high quality data. Okay? Because that is the key for us to understand, interpret, and find the trajectory of different markers which can lead us to develop a diagnostic biomarker. That's the goal. And the cause of all the diseases, of course, have the root in the molecular and cellular mechanisms. So we need the science behind what this disease, how this disease progresses, how does this disease get triggered, and so on and so forth. So there is an understanding of that mechanism is very important. And of course, we need to do the translational part of development of the biomarkers. So first we need to identify, we need the science to identify what biomarkers are relevant and how do they progress. And then, of course, develop intervention. So the data collection part, there is a research infrastructure that is needed. I'll quickly go through what we do in this study, but we use machines like optical coherence tomography to see the condition of the eye. We do genome sequencing, we do GWAS, we have EEGs, so we do have Simoa machine looks at the protein concentrations in blood, confocal microscopy, we look at tissues, weight, and so on and so forth. We store everything in a biobank, and then there is a audiometry, vascular, and so on and so forth. Then we use the IISc's 3 Tesla MRI machine and the animal facility for doing research. So what do we do in the longitudinal cohort? We have 2 cohorts. An urban cohort of over 1,000 subjects started in 2015 called the Data Longitude Study on Aging, and the rural cohort started in 2018 for 10,000 subjects called the SANSCOM, funded by 2 trusts. And this is the first of its kind longitudinal, large-scale longitudinal study to study the risk factors of dementia. So what exactly happens? There is a awareness camp we hold. We get participants to volunteer. This is because these are all volunteers. Then we use different mechanisms of engaging with them. What we do is basically collect information about the socio-demographic information on the right side, the clinical assessment, their medical history. Then we do a cognitive test to understand what is the cognitive score. And then we take blood, we do whole genome analysis, we go through MRI, audiometry, and so on and so forth. So all this data comes through. So far we have been able to collect data in a longitudinal manner. Remember that the disease is progressive, so we need to understand how different parameters are progressing in time. So we take healthy subjects about the age of 45, they participate, and then we have so far been able to, in Bangalore city, if you're above the age of 45, you can enroll into our cohort. We have been able to do that. In the rural, we have very ambitiously embarked on 10,000, so we are likely to touch the 10,000 this year, but that will be the first visit of the 10,000 people. We then need to do a longitudinal study over the years. Of course, there are challenges because the subjects are not able to understand the research aspect. They have other commitments. They're all volunteers. We can't force them. We can't incentivize them with too much money because of ethical reasons and so on and so forth. Anyway, so the other project where we generate a lot of data is the Genome India project funded by the government, where we have done an analysis of 10,000 genome sequences have been done for a population from 20 different institutions. People have collected blood samples from 99 ethnic groups of India. Samples have been collected with an average number of 100 per ethnic group. And this is going to, the collection has happened, the sequencing has happened, data analysis is in progress. So what do we do from a data point of view? As you can notice, that we have so many different sensors and devices to generate data. Each one of them will generate huge amounts of data per volunteer. So we have 10,000 volunteers, 1,000 volunteers, and 10,000 genome sequences, and so on and so forth. So all this flows into a system, which is a high performance computing storage and computing system. and we are in the process of trying to create a platform which is secure, right? Enabled, maybe federated scientific data sharing platform. The architecture I'll skip. But the goal is now to see whether we can leverage the tools of AI to understand the patterns in the data we have collected so far. So that's the goal, and we are trying to see whether we can do something meaningful to detect the early onset of dementia. So I'll just skip all this. It is just to, we have acknowledgement from different agencies. Thank you.
Any questions for Dr. Hari?
Yes. So when you're going to release the data for us to analyze? Good question.
Yes, we want to know that too.
Yeah. So right now we are in the process of curating the data until the data set achieves its objective, because we need longitudinal data. There is no point releasing the first round of data. We are doing a longitudinal study of 10 to 15 years, right? At least. So we need a few number of follow-up visits where we collect all this data, and then we will be in a position to release it after the analysis. The structure is for releasing it for open public.
Aware of the timeline, you can shed some light on that.
Timeline will be not less than 5 years, because the visits are, for the rural we collect data every 2 years. So there's no point in releasing only one sample or two samples of the subject. So we need to collect data. So we need to do that. But urban we have multiple, but not a large number. For statistical analysis, we need enough number for the test, your hypothesis testing to be strong. So it will take time.